Hello everyone. As I promised last time, I'm going to continue with those theoretical topics and lectures in English for our international students. And if you remember, we started with the metabolism of carbohydrates and with the topic digestion and absorption of carbs. So now we're going to move on to see which are the upcoming catabolic pathways of carbohydrates and especially glucose. And the first one on the menu is going to be glycolysis. So let's start. So glycolysis is a metabolic process which is the first catabolic process of glucose which is obtained upon the digestion of complex carbohydrates. And predominantly all other monosaccharides can be converted to glucose following specific patterns and following specific pathways, and I'm going to talk about that in one of the upcoming lectures. So we can conclude that definitely glucose has a central position in carbs metabolism. So there are a few important catabolic pathways glucose undergoes, or pathways of degradation. So definitely it's glycolysis, which is going to be our today's topic, followed by oxidative decarboxylation and Krebs cycle, which are going to be our upcoming topics. So all of these processes actually yields the final formation of carbon dioxide and water from the molecule of glucose. And during this degradation pathways, certain amount of energy is released and captured in the formation of ATP. So the first catabolic pathway is glycolysis, which actually serves to convert glucose molecule to pyruvate when we are talking about aerobic conditions, which means in the presence of oxygen, and to L-lactate in anaerobic conditions when we are talking about processes which occurs in the absence of oxygen. So anyway, in order for the glycolysis to start, glucose has to enter the cell, because so far we mentioned that glucose and other monosaccharides are released from enterocytes into portal bloodstream and from liver into systemic circulation. So glucose is now available to tissue cells, but first it has to get in. So what about just a few words about the entrance of glucose in cells? So despite the fact that there is a certain concentration gradient between the extracellular and intracellular compartment, meaning that after a carb-rich diet and a carb-rich meal, the concentration of glucose is significantly higher in the blood than inside the cell, it cannot diffuse directly. So the mechanism which is employed for this purpose is facilitated diffusion. And as you know from the general part of biochemistry, there is a family of glucose transporters located in a cell membrane required and needed for such a transport. They are also marked as GLUT transporters as a short and abbreviation for glucose, GLU, and transporters as T. So there are 5 types of glucose transporters, and predominantly they are tissue specific. I'm going to give you just one example. For example, glucose 4 transporter is located in adipose tissue and skeletal muscles. All of these transporters are coordinated, activated, or inhibited upon the action of insulin and anti-insular hormones. So a little bit more detailed story about those glucose transporters will be delivered to you upon the story on hormones, insulin, and glucagon. So once glucose is in the cell, it can undergo and enter the glycolysis process. So you will see, because this is the first metabolic pathway that we're going to talk about. And now it's the right moment to set the patterns. So whenever we are going to start with one specific metabolic pathway, the first fact that we're going to mention is where does that metabolic pathway takes place. So in this case, glycolysis occurs incomplete in cytosol, And of course, like all other pathways, it consists of several reactions forming several intermediary products. So let's start from the beginning. The first reaction is conversion of glucose to glucose 6-phosphate, and this is the reaction of phosphorylation. So one phosphate group is attached at the position number 6 and the molecule of glucose. You can see the reaction on the slide. So what can we say about this reaction? And we're going to follow the same pattern not only for glycolysis and the rest of the reactions, but if you catch up with the correct way and pattern how to explain the pathway, you can apply that knowledge and those tips and tricks to other pathways as well. So let's start from the beginning. When we say the term phosphorylation, we mean addition of phosphate group. So the question is, who is the donor of that phosphate group? You can remember that in biochem pathways, the donor of phosphate group is ATP. There are going to be just a few reactions, maybe two, one in this glycolytic pathway, one in Krebs cycle, where the donor of phosphate group is going to be inorganic phosphate. In all other cases and situations, the donor of phosphate group is ATP. So you can remember that there is always ATP involved in the reaction whenever we have a phosphorylation. Why here? I'm sorry. So anyway, so we can conclude that one high energy bond is consumed during this reaction, and we have to have this in mind in order to be able to calculate the energy yield of this metabolic process at the very end. What else can we say about this reaction? That this reaction is catalyzed by the enzyme hexokinase. How can you remember the names of enzymes and to avoid learning their names by heart? So you can apply some kind of logical thinking, at least a little bit. So if you remember our general part of biochemistry, we mentioned that the group of enzymes which belong to the group of transferases, which are responsible for transfer of phosphate group from one molecule to another, are marked as kinases. So in this case as well, we have the transport of one phosphate group from one molecule to another, so it's going to be a kinase. Why hexokinase? Because glucose is a hexose. So hexoses are actually phosphorylated by the enzyme hexokinase. And of course we need activators, and how to memorize that magnesium ions are activators. I'm going to just give you one tip, that magnesium is an required activator for almost 300 reactions in our body. So if you say that you need magnesium ions, probably you won't be wrong. So this is a rather fast reaction, and it takes place even if the concentration of available glucose is rather low. So what else can we say about this reaction and about this enzyme? So this enzyme hexokinase is one of the 3 regulatory or rate limiting enzymes of the glycolytic pathway, and now is the moment to explain what does that rate limiting means. That means that the rate of the overall metabolic pathway depends on the activity of that specific regulatory enzyme. So we're going to have at least one regulatory or rate limiting enzyme for each metabolic pathway. So now we are introducing some new terms, and we should memorize them for the rest of the metabolism during biochem course. Also, something that you have already done in our general biochemistry section is that it's allosteric enzyme, which means that glucose 6-phosphate as a final product of this reaction is negative allosteric modulator. So this reaction undergoes feedback inhibition upon the presence of the excess amounts of glucose 6-phosphate in a molecule. So this enzyme is also not absolutely specific for glucose, so I have already mentioned that it's able to phosphorylate other hexoses as well, and that is the reason why we have the name hexokinase. But that is so interesting because in liver cells we have the same enzyme. We can even say isoenzyme form of hexokinase, which is called glucokinase. And there are just very small amounts of hexokinase found in hepatocytes. Why is that so important? We mentioned that isoenzymes actually catalyze the same chemical reaction. It's not a question here as well. So both enzymes or isoenzymes catalyze phosphorylation of glucose at the position 6. But they differ in some other physicochemical properties, kinetic properties, and so on. So we're going to focus on those kinetic properties. So they differ in Michaelis constant for glucose, meaning they differ in affinity to bind glucose and phosphorylate glucose. So hexokinase exhibit rather low Km, which means, if you remember, that it exhibits rather high affinity. What does that mean? That means that this enzyme can phosphorylate glucose even when just small amount of glucose is available. So you need a rather low concentration of glucose present in order to have the activity of hexokinase. Glucokinase, on the other side, has significantly higher Km, which means significantly lower affinity, which means that this enzyme needs to have a lot of glucose available in order to start phosphorylating it. So let's conclude. Hexokinase phosphorylates almost total amount of glucose which enters the cell, which is important for maintaining high concentration gradient of glucose between blood and intracellular compartment. It also enables, which is of crucial importance, the sufficient tissue supply with glucose even when the concentration of blood glucose is rather low. On the other side, glucokinase will catalyze the phosphorylation only when the amount of glucose available is high, for example, after the carb-rich meal. These differences are so important for maintaining the proper glucose distribution in the body. So both enzymes exhibit significant effects on glucose levels in the body and its distribution between blood and cells. So for example, when glucose concentration in blood is low, which happens between meals or during fasting or performing some carb-free diets like keto diets and so on. So the concentration of glucose will be low. Under these conditions, and having in mind the previous statements, liver or hepatocytes will not uptake significant amounts of glucose. So glucose will remain available for consumption to other tissues by hexokinase. On the other side, after the carb-rich meal, the levels of glucose go up. So what happens? There is a lot of glucose available. So glucokinase is going to be activated and its activity becomes significant. So the liver is going to uptake significant amounts of glucose. Why is that so important? Because liver is one of the tissues apart from skeletal muscles which is capable to store the excess amounts of glucose in the form of glycogen. Or it can use that glucose for the biosynthesis of other compounds like 3-acylglycerols, fatty acids, and so on. We will come to it later on. So the second reaction is glucose 6-phosphate conversion to fructose 6-phosphate, and this is one typical reaction of isomerization. So if you see just glucose 6-phosphate is converted to fructose 6-phosphate, and please notice that this reaction is reversible oppositely to the previous one, and this is again the right moment to emphasize the fact when we say that the reaction is reversible, and to explain what does that mean. There is one other metabolic pathway that we are going to talk about later on, which actually synthesizes glucose from simple compounds like lactate and pyruvate and some other compounds, and that pathway is the reversible pathway in most of its steps to glycolysis, and that pathway is called gluconeogenesis. So when explaining glycolysis, whenever we say that the specific reaction is reversible, remember that the same reaction will occur in gluconeogenesis. And in those couple of reactions, when the reaction is irreversible, we're going to have some other enzymes involved in gluconeogenic process. So that is what does that mean, reversible reaction. This reaction is catalyzed by phosphoglucose isomerase. So again, the name of the enzyme is pretty simple and logic. So there is isomerization, so it has to be isomerase enzyme. And who is isomerized? Glucose 6-phosphate, so phosphoglucose is isomerized. No other specificities regarding this second reaction, so we can move on to the third one. The third reaction of glycolysis is conversion, or phosphorylation of fructose 6-phosphate to fructose 1,6-D-phosphate, and this is the phosphorylation at the position number 1 as it's presented here on a figure. So let's start again with the same idea from the reaction number 1. Again, when we have phosphorylation, then we know that the donor of the phosphate group is ATP. What else should we know? That if ATP is used for phosphorylation, we are actually consuming another one, the second out of the second high energy bond for this reaction, converting it to ADP. The enzyme which catalyzed this reaction is phosphofructokinase 1, which means it phosphorylates already phosphorylated fructose at the position number 1. What is important regarding this enzyme? This is the main rate limiting enzyme of the glycolytic pathway, and it's the second out of 3 regulatory enzymes. So actually we can conclude that the overall rate of glycolysis in general depends on the enzyme on this enzyme activity. And let's move on to the fourth reaction. Now we have one breaking of the molecule, 6-carbon molecule into 2 3-carbon molecules. So fructose 1,6-D-phosphate is broken down to glyceraldehyde 3-phosphate, GAP, and dehydroxyacetone phosphate, DHAP, and here is the reaction of hydrolysis. So I have written this reaction with fructose 1,6-D-phosphate in the form of an open chain, just to be easier to follow the breaking into 2 3-carbon molecules. So it is also the reversible reaction, this time catalyzed by the enzyme aldolase. And if you're asking why aldolase, why is the name given aldolase, remember our organic chemistry course when we mentioned the reaction of aldol addition, and this is the reversible aldol addition. So that is the reason why the name of the enzyme is aldolase. So what is interesting regarding this reaction? The predominant product of this reaction, more than 90%, is DHAP, dehydroxyacetone phosphate. But you will see from the from the next slide, the next steps, that only GAP is consumed in further steps of glycolysis. So we have to have one more additional step on glycolysis, which is gonna be used for conversion of dihydroxyacetone phosphate into GAP. So this is the reaction of isomerization, and here it is written again. We have a reversible reaction, so the same will occur in gluconeogenesis. And the enzyme which catalyzes this reaction is triose phosphate isomerase, which means it isomerizes one triose phosphate into another triose phosphate, one ketose into one aldose. So glyceraldehyde 3-phosphate is the one which undergoes further changes in glycolytic pathways, which actually brings us to the conclusion that from this point on, reactions 6, 7, 8, 9, and 10 are actually gonna happen two times, twofold, with two molecules formed from one fructose 1,6-D-phosphate. So coefficients in the upcoming reactions will be multiplied by 2, and we have to have this in mind when calculating the energy yield of the overall pathway. So let's see what's going on in the reaction number 6. In the reaction number 6, glyceraldehyde 3-phosphate is phosphorylated and oxidated to 1,3-D-phosphoglycerate. So here is the reaction. The reaction is reversible, so the same occurs in gluconeogenesis. And regarding the fact that it's oxidation and phosphorylation at the same time, again, we can set the name for an enzyme, which is going to be dehydrogenase. Remember that oxidoreductases, which are used for oxidation and redox processes but requiring coenzyme, are marked as dehydrogenases. Notice that this time, and this is an exception that I've mentioned previously, the donor of the phosphate group is inorganic phosphate and not the ATP, so be careful about this step. So the enzyme is glyceraldehyde 3-phosphate dehydrogenase because it's gonna oxidate glyceraldehyde 3-phosphate into 1,3-D-phosphoglycerate. What else do we know? Whenever we have a dehydrogenase, we need a coenzyme. Common coenzymes of oxidoreductases are predominantly NADH, but they can be FAD or FMN. So in this case, one molecule of NAD plus is reduced to NADH reduced form, which is going to be also very important when calculating the energy yield due to the fact that NADH can enter the electron transport system later on. What else do we know? From the general biochemistry, we know that 1,3-D-phosphoglycerate is a high energy compound. And again, one more pattern. Whenever you have a formation of a high energy compound during certain metabolic pathway, the upcoming reaction is going to be its hydrolysis and the release of energy and the synthesis of 1 ATP that you will see in a couple of seconds. So the reaction number 7 is what I've told you. High energy phosphoester bond is hydrolyzed, the energy is released and captured in the form of 1 ATP molecule. So this is also the reversible reaction, so the same takes place in gluconeogenic pathway, and in this case enzyme phosphoglycerate kinase is involved in catalyzing this reaction. So, as I've already mentioned, energy is released and one molecule of ATP synthesized, but at the point when we're going to calculate the energy yield, we're going to multiply all this with 2, so we're going to have the synthesis of actually 2 molecules of ATP, so be aware of this. And this is an example of phosphorylation at the substrate level that you talked about in our general biochemistry course. Now, this is an application in practice in metabolism. So the reaction number 8 is conversion of 3-phosphoglycerate 3-PG into 2-phosphoglycerate 2-PG. So this is the reaction of mutation, and it's also the reversible reaction. Here it is. 3-phosphoglycerate to 2-phosphoglycerate, and phosphoglycerate mutase is the responsible enzyme. But despite the fact that this reaction looks quite simple, so what do we need to do just to convert phosphate group from the third to the second carbon atom? But actually it's not that simple, and usually it's not that simple in biochemistry. So it's not a simple transfer of phosphate group, but rather 3-PG is bound to the enzyme, to its active site, and then in the active site, in the active center, you have one phosphate group. So when substrate is attached to the active site, actually it's gonna grab and attach to that phosphate group as well. So one intermediate named 2,3-D-phosphoglycerate is gonna be formed. After that, a product is gonna be released from the enzyme substrate complex, but this time our molecule is gonna keep the phosphate group at the position number 2, and the phosphate group which was at the position number 3 is gonna be left on the enzyme. So the enzyme is gonna be regenerated in the form it was at the beginning of this reaction, and 2-PG is gonna be produced. This is very interesting reaction, and I'm gonna ask you, have you ever heard before about 2,3-D-phosphoglycerate? I think this is not the first time that you hear about this molecule. So where did you and when did you hear about this? Good point. It has to do something with hemoglobin, with erythrocytes, and with the affinity of hemoglobin to bind oxygen. And at the time we mentioned that 2,3-D-phosphoglycerate actually lowers the affinity of hemoglobin for oxygen, thus increasing the release of oxygen to tissue. So in erythrocytes, the concentration of 2,3-D-phosphoglycerate is significantly higher than in other cells. Why is that so? Because erythrocytes are very specific cells, not having nucleus, but more important, not having mitochondria. So no oxidative metabolism occur in erythrocytes. So only anaerobic glycolysis is actually the only possible energy yielding pathway for erythrocytes, which means that the anaerobic part of glycolysis is gonna happen with the high rate, consuming a huge amounts of glucose, is thus producing a lot of 2,3-D-phosphoglycerate, which is gonna affect hemoglobin from erythrocytes to lower the affinity to bind oxygen and to achieve the easier release of oxygen to tissues. So obviously erythrocytes need more 2,3-D-phosphoglycerate than any other somatic cells. So there are two more enzymes which are active here. D-phosphoglycerate mutase, which is responsible for the synthesis of 2,3-DPG, and 2,3-DPG phosphatase, which degrades or hydrolyzes 2,3-DPG. So this is considered like a short additional pathway of glycolysis. But let's get back to our glycolytic pathways. So the ninth reaction, 2-phosphoglycerate is converted to phosphoenol pyruvate, followed by the release of one molecule of water. So the reaction is reversible, catalyzed by the enzyme of enolase. And again, if you take a look at the product formed and having in mind your general biochemistry knowledge, you will notice that phosphoenol pyruvate is again a high energy compound. Actually, this is the high energy compound containing the highest amount of energy. So it's at the top position in on that high energy levels, high energy compounds list. So, and like we already mentioned, whenever you have a high energy compound form, the upcoming reaction is going to be its hydrolysis and the release of energy. So, and that is the final and the last reaction of aerobic glycolysis. So, phosphoenol pyruvate is hydrolyzed to pyruvate. The energy is released and captured in the form of ATP. So again, because we are dealing with phosphate groups and ADPs and ATPs, we have a kinase enzyme, so it's going to be pyruvate kinase, which is activated by the presence of magnesium and potassium ions. And it's the third and the last regulatory enzyme of the glycolytic pathway. And again, like previously, we have the example of the phosphorylation at the substrate level. So we can conclude that the final products of aerobic glycolysis are, so pyruvate definitely, then ATP and NADH. But in anaerobic conditions, so without the oxygen present, we have one more step, 11th step, which is the conversion or by means reduction of pyruvate to L-lactate, reaction catalyzed by famous lactate dehydrogenase, and it was our topic previously, if you remember. Like all other dehydrogenases, it requires the presence of an adequate coenzyme, and again, it's going to be NADH. This NADH coenzyme is going to be the one which is produced in the aerobic part of the glycolysis. So in anaerobic glycolysis, NADH produced is going to be consumed, so no net formation of NADH coenzyme. This enzyme is also absolutely specific for L-lactate, so D-lactate is not formed in this case. So we can say that the final products of anaerobic glycolysis are L-lactate and ATP. So please remember this. We can now speak a little bit about L-lactate because anaerobic glycolysis is especially intensive in erythrocytes, because I have told you that this is the only energy yielding metabolic pathway in erythrocytes because they lack mitochondria. So no aerobic metabolism, no further oxidative degradation of glucose, no oxidative degradation of lipids. So actually, erythrocytes are consuming and degrading an enormous amounts of glucose, thus significant amounts of L-lactate are formed, and L-lactate, which is formed, is primarily transported to the liver, where it enters the glucose resynthesis, gluconeogenesis. First it's converted to pyruvate, and then pyruvate is converted to phosphoenol pyruvate via oxaloacetate, but this is going to be one separate topic in the upcoming days. So let's calculate the energy yield of glycolysis. So first let's do with aerobic glycolysis. So the energy in the form of ATP is consumed or gained in the following reactions. So if you remember, it was the first reaction when we used up 1 ATP, so it's going to be minus 1 ATP. In the reaction number 3, again, we used 1 ATP for phosphorylation of fructose, so we're minus 2 so far. But in the upcoming reactions, we have the formation of NADH coenzyme, but remember, we multiplied coefficients with 2. And remember that in electron transport system, 1 NADH gives 3 ATP, which gives us the total of 6 ATPs. Then we have the formation of 1 ATP in 1,3-D-phosphoglycerate hydrolytic reaction, so multiplied with 2, it's going to be 2 ATPs, and formation or hydrolysis of phosphoenol pyruvate to pyruvate yields the formation of 1 ATP, which gives us 2 ATPs when multiplied with 2. So when we do a little bit of calculus, 6 plus 2 plus 2 minus 1, we get that the total energy yield of aerobic glycolysis is 8 ATPs. What about anaerobic glycolysis? the energy is consumed or gained in the same way as in aerobic glycolysis, but we have the additional consumption of NADH coenzyme in LDH reaction. So no NADH formed in this case, so minus those 6 ATPs gives us the energy yield of anaerobic glycolysis is only 2 ATPs. So let's move on to glycolytic pathway regulation, and this is going to be the last part of all our stories on metabolic pathways. So when we're going to see how those pathways are regulated and which factors affect the rate of that metabolic pathway. So if you remember, we emphasized that there are 3 enzymes, hexokinase, phosphofructokinase, and pyruvate kinase. So there are a lot of factors which affect the activity of these enzymes. First, those are hormones as main regulatory molecules in our body. So insulin is the one which stimulates the activity of these enzymes, and glucagon and other anti-insular hormones are responsible to inhibit enzyme activities. But also, apart from the hormonal regulation, each regulatory enzyme can be affected or its activity regulated by the action of some different molecules, not only products of the specific reaction, but intermediates of that or some other pathways. So hexokinase activity is inhibited by glucose 6-phosphate by the terms, if you remember, we mentioned by the terms of feedback inhibition. Phosphofructokinase is affected in the way that it's activated by ADP, AMP as markers of energy deficit in a cell, also fructose 2,6-D-phosphate, ammonium ions, and phosphate ions. But the action of this enzyme is inhibited by ATP and citrate because those molecules are markers of energy sufficiency in the cell. And finally, pyruvate kinase is activated by fructose 1,6-D-phosphate, which is one of the intermediates of glycolytic pathway, and again the ATP as a marker of sufficient energy in a cell inhibits the enzyme activity and slows down the glycolytic pathway. So actually that would be it regarding the first metabolic pathway, with a lot of tips and tricks how to memorize all these facts, which pattern to follow to be able to understand these pathways easily and to memorize the bunch of enzymes and intermediates easier than memorizing them by heart. So I hope this was understandable. I hope that you're okay with this topic, and next time when we meet here online, we're gonna talk about the fate of pyruvate and its upcoming degradation. So till then, I'm gonna say goodbye for now. Have a nice rest of the day, and bye bye.